and behavior in chapter 2 of the textbook. Before I do that, I want to read you from a book that I'm reading right now. It's called The Myth of Artificial Intelligence: Why Computers Can't Think the Way That We Do by Eric J. Larson. This is on page 250. He's talking about the human brain. He says the human brain has more than 100 trillion neuronal synapses. So a computer recording a sample binary piece of information about synapses, such as whether it fires in a time window or not, would require 100 terabytes. The amount of storage needed to store even this very simple information every second over the course of one day for one person would more than 100,000 terabytes or 100 petabytes. Supercomputers these days hold about 10 petabytes. And this quick calculation doesn't account for the changes in connectivity and positioning of these synapses occurring over time. Counting how these connections change just after a good night's sleep or a class in mathematics amounts to a whopping figure and many more bytes than the estimated 1,080 atoms in the universe. So, as you can see, artificial intelligence, and we're going to talk about this in the 321 class, and that's the reason I'm reading the book is for the 321 class. The, what's my class? Cinema therapy, social media, and alternative platforms, the gaming platforms. So that's artificial intelligence is part of that. As you can see, according to Mr. Larson, our own brains, the human brain, no matter what your what your intellectual capability is far more intelligent because it can think in a lot of different directions at the same time. Most artificial intelligence can only think in one direction at a time. And it's whatever it's been programmed to do. It can only think in that direction. And it can't come up with novel ideas. Computers don't come up with novel ideas. They can only repeat what they've been told. So my point is that the human nervous system is quite complex, and we're going to talk about it right now. The 100 million synapses, 100 trillion, is that what it was? 250, wait a second. Did I mess the number up? More than 100 trillion neuronal synapses, 100 trillion. It's a lot because a trillion is a million million. And 100 million million, well, that's a lot anyway. So the human brain is quite complex. And that's one of the things that I want you to be aware of. So let's go ahead and get started. The nervous system and behavior, chapter 2. The nervous system is composed of cells called nerve cells or neurons. The average human body has between 100 billion and 150 billion neurons. That's the neurons. Now, if we talk about the synapses, some of them have as many as 150,000 synapses each neuron. So we can just can keep compounding this number until we come up with an astronomical number like 100 trillion synapses. Neurons communicate with each other at a point known as a synapse. Another cell that protects and adds structure to the nervous system is the glial cell. A neuron is composed of 4 major parts. The dendrites collect the stimuli or information and transmits it to the rest of the neuron. The soma or cell body contains the nucleus and serves to maintain the cell. The axon is the main transmitting apparatus. The axon terminals are where the neuron passes on the information to other cells or neurons. Neurons can be classified in 3 basic ways. They can be classified by shape, by size, or by function. Multipolar neurons have many dendrites and a single axon. Most of the neurons of a vertebrate brain are multipolar, go in different directions at the same time. Bipolar neurons have a single dendrite at each end and a single axon at the other. Most of the neurons of the sensory system are bipolar, especially those of the retina and the olfactory system. Monopolar neurons have a single branch coming off the soma that branches in two different directions. One branch will be the input direction and the other will be the output direction, but there are axons going in both directions. Some neurons are very large and some are very small. Examples of small cells would be granule, spindle, and stellate neurons. Examples of large neurons would be pyramidal, Golgi, and Purkinje neurons. Functionally, neurons either receive stimuli, create a response, or connect the two. Sensory neurons collect the stimulus and pass the message on to the vast network of connector neurons or interneurons. Motor neurons connect to muscles or glands and create the response by making the muscles contract or by changing the activity of the gland. Neurons do not touch each other, but communicate between the dendrite of one neuron and the axon of another across a gap known as the synaptic cleft. At the end of the axon is the axon terminal. The axons end in expanded areas called buttons or boutons, which is actually French for buttons. Bouton. The end of the axon is known as the presynaptic zone. The area of the receiving dendrite is known as the postsynaptic zone. The message is actually transported by a chemical known as a neurotransmitter that is contained in spheres in the axons called synaptic vesicles. You can see the synaptic vesicles here. This is the presynaptic zone. This is the postsynaptic zone, and this is the synaptic cleft. And in order for a response to take place, the synaptic vesicles need to release the neurotransmitter, and it will cross the synaptic cleft, and it will be received by the postsynaptic zone by receptors. Now the receptors are really important because the number of receptors will tell the neuron exactly how much response there should be. So if we're dealing with, let's say we're dealing with an illicit drug. Let's talk about which one do we want to talk about? Let's say we're dealing with marijuana. Marijuana is crossing the synaptic cleft. Now let's use something better than that. Let's use heroin. Heroin is crossing the synaptic cleft. Well, heroin initially, of course, there will be lots of receptors on the other side that the heroin can find the receptors, and it will make a huge impact on the individual. Well, because the heroin is toxic, because the heroin is fairly destructive, the neuron will try to protect itself, and it will protect itself by reducing the number of receptors so that the next time the heroin is in the synaptic cleft, the response won't be as extreme and it won't potentially destroy itself. All the neuron is trying to do is protect itself. It's trying to reach a point of balance. And balance in this case is no heroin. So it tries to control the response to the heroin by downregulating the number of receptor sites. I hope that makes sense. Now let's say that the guy hasn't used heroin for 2 months and his body is starting to crave heroin. It wants more heroin. So what will happen is the number of receptors will actually increase, and that's an upregulation. That's known as an upregulation. And that's what causes the craving. One of the interesting areas of study dealing with the neuron are projections coming from the dendrites called spines. These spines seem to change with experience and have been known to change within seconds when an individual is exposed to a novel stimuli. And that's what the spines look like. These are, and they will have receptor sites on them, and these can change very rapidly. The axon projects from the soma in an expanded area known as the axon hillock. The electrical impulse begins in the widened hillock and intensifies as it moves into the narrower axon proper. Almost all neurons have only one axon. However, some have axons that divide and form several branches. This branching is known as axon collaterals. When someone smashes their finger, the stimuli must follow a nerve pathway to tell the brain what has happened. The nerves that take the stimuli from its point of origin to the central nervous system are called afferent neurons. When the brain or spinal cord sends back a message to the affected area, the neurons on the reply pathway are referred to as efferent neurons. So it's afferent neurons take the message to the central nervous system, and efferent neurons take the message back to the area. And in the case of the smashed finger, what will happen is the afferent neurons will tell the brain that it has a smashed finger, and the efferent neurons will tell the individual to move their finger so it doesn't get smashed again. The soma manufactures proteins that maintain growth and function in the neuron. This protein is able to make its way down into the distant areas of the axon and dendrites by a process known as axonal transport. The protein is moved along tiny hollow cylinders known as microtubules. Protein molecules travel down smaller rods inside the microtubules called neurofilaments and even tinier microfilaments. Now this is important because a lot of pharmaceuticals will use this method to get from one place to another. So they will use the microtubules and the microfilaments and the neurofilaments in order to get from one place to the other. And that will be the reason that the pharmaceutical works. Glial cells are part of the neuronal system in that they provide structure to the neuronal groupings and help hold them in place, and they help maintain the neurons by feeding them. There are 4 types of glial cells: astrocytes, microglial cells, oligodendrocytes, and Schwann cells. Astrocytes are star-shaped glial cells, and that's why it's called astro. Astro means star in Latin. They are star-shaped glial cells that protect neuronal bundles, especially in the brain. The dura mater, a tough sheet that is wrapped around the brain for protection, is composed of astrocytes. Astrocytes contribute to the metabolism of the neuron and provide restorative protein to them. Microglial cells are very small, hence the name micro. They migrate to areas of injury or disease and remove debris from injured or dead neurons. Now this is really important because as people age, their ability to remove dead debris and dead neurons decreases. And if this decreases too much, it will cause a disease known as Alzheimer's disease. So the microglial cells are very important. Oligodendrocytes that are the myelin sheaths that surround and protect the neurons are created by glial cells. These myelin sheaths not only protect the neuron, but accelerate the response of the neuron. Oligodendrocytes provide the myelin in the brain and the spinal cord. Oligodendrocytes means little tree cell, and that's because it is so involved. It's the largest of the oligodendrocytes and the Schwann cells are the largest of the glial cells. Schwann cells construct the myelin outside the brain and spinal column. Schwann cells are small, and each one will wrap itself around the axon to protect it and accelerate response. When you look at the myelin formed around an axon, you will see that Schwann cells don't touch each other but leave a gap between each cell. These gaps are known as nodes of Ranvier. When an individual suffers a blow to the head, the injury tends to heal slower than a blow to any other part of the body. This is because, besides causing localized damage, the glial cells become larger by taking on moisture. This is known as edema, and it may put pressure on the select parts of the brain, causing malfunction. But the reason it's there is to protect the brain. And the glial cells are just trying to make sure that the brain doesn't get damaged any more than it already is. And that's why it swells up. It swells up to protect it. So if you sprained your wrist would swell up. And the reason it swells up is to immobilize it and to protect it from another injury. And that's why you have so much swelling in your body. It is a defense mechanism. It's trying to protect you. It's not trying to hurt you. It's trying to protect you. And this is one of the reasons why if you break a bone, what normally will happen is they splint it and then they'll tell you to come back. They will put a cast on it after the swelling goes down. That's what they say. Why do you have the swelling? The body is trying to protect itself by swelling. And this is known as edema. It will send moisture into the area to surround the area to make it hard so that you can't move the wrist anymore. And that's what the swelling is all about. The nervous system of the human body can be subdivided into two nervous systems. The central nervous system is comprised of the brain and the spinal cord. The peripheral nervous system includes all other neurons outside the brain and the spinal cord. There's a peripheral nervous system, and there is the central nervous system. The peripheral nerves are categorized by what part of the central nervous system they are connected to. Cranial nerves are connected directly to the brain. Spinal nerves are connected directly to the spinal column. Autonomic nerves in the autonomic nervous system are connected to glands and organs and trigger automatic responses from these areas. There are 12 cranial nerves that control sensory or muscular movements in the head or neck region. 4 of the nerves control the muscles of the eye and eyesight sensations. 4 control the muscles of the neck, the face, and tongue, and the sensations of tongue, sinuses, and throat. 1 controls the sensation of the inner ear. The vagus nerve controls the heart, lungs, and digestion. There are 31 pairs of spinal nerves, 1 pair on each side of the body. There are 5 segments of the spinal cord and column. There are 8 cervical nerves, that's in your neck. Cervical means collar, and that's around the neck. There are 12 thoracic nerves, and that is your torso. Thoracic means brace, and so it would be around your torso. There are 5 lumbar nerves, and that is your lower back. And lumbar means belt, so that's why they call it lumbar nerves. And this, of course, is where most people damage themselves. The lumbar region of the back is very sensitive. The reason it's so sensitive is because it's not protected by something else. It's kind of free moving and free flowing. If you look at the thoracic region, you've got your rib cage. It's where all of your organs are. The reason your rib cage is there is to protect all these vital organs. It needs to be there so that somebody can't just punch your heart. The cervical nerves are controlled. Well, the cervical nerves really don't have a lot of protection, but we tend to protect our heads. The one region of our bodies that we really don't protect is the lumbar region. And of course, when we lift something, we lift with our backs. We lift with our lumbar region, unfortunately. And that's why so many people have lower back problems. There are 5 sacral nerves, that's in the pelvic girdle. And of course, you've got the pelvis there to protect those nerves and that area of your body. And then there's one coccygeal nerve, and that is your tailbone. That's where your butt is. There, got it. All right. And there are all of your nerves. This is the area. This area right here. You can see that the rib cage protects you. Your arms are right there. Your pelvis protects you here. This area really doesn't have a lot of protection, but we tend to focus on our heads, and for that reason we protect the cervical nerves. But there's just nothing here to protect you between your pelvis and your rib cage. That is normally where people injure themselves, in the lumbar region. The autonomic or automatic nervous system is divided into two nervous systems. The sympathetic nervous system prepares the body for action by shutting down normal functions, increasing power stores, and increasing sensory activity. The parasympathetic nervous system returns the body to a normal state after sympathetic reactions. When the need for action is detected, the sympathetic nervous system will dilate your pupils, improving your eyesight, inhibit salivation, causing dry mouth. It will relax your airways, allowing deeper breathing. It will accelerate your heart, stimulate sweat glands, stimulate liver to release energy stores. It will slow your digestion because that takes away too much of your energy. It will inhibit kidney function. It will constrict blood vessels in the skin, making you pale, and now you're either ready to fight or flee. The parasympathetic nervous system constricts your pupils, it stimulates salivation, it constricts your airways, it slows your heartbeat, stimulates digestion, dilates blood vessels in the intestines, and dilates blood vessels in the skin, making you flushed. So once the danger is gone, then all of these things take place. And if you've ever been in a situation like that, then you know that this is a really good feeling. When all of a sudden you can taste your mouth again instead of having dry mouth. Okay, your heart's not beating as strong as it was before. Once upon a time, when I was in college, I had a situation where my sympathetic nervous system kicked in. And the reason it kicked in was because I was fighting with my wife. I had student teaching on Monday morning, and then I had a track meet in the afternoon. So what happened to me was that because of the fact that I was fighting with my wife and she demanded to go home, and I took her halfway home, and then she decided she didn't want to go home anymore. So we were fighting all weekend. So this was I got all excited about her demanding to go home and whatnot. And then the next day, of course, I had student teaching, and teaching is not easy, and we hadn't really been trained to teach, and here I was trying to interact with the students. And then later on that day, I had a track meet. So at the track meet, because my sympathetic nervous system had been firing all weekend and firing that morning, and because track meets are quite exciting, I had all of these things happen to me. All of these things happened to me, and the one thing one problem that I had was my kidney functions had slowed down to the point of stopping, and this had been going on from Friday. Friday, my wife was giving me a hard time. Saturday, Sundays when she decided she had to leave, and I started to take her back, and then she decided, oh, maybe things aren't so bad. So then she came back, and then on Monday, of course, I had to student teach all morning. I had to be there at 8 o'clock, and I student taught until 12 o'clock, and then the track meet was at 4. So here I was, Friday, Saturday, Sunday, Monday, 4 days of my sympathetic nervous system taking hold of my body. And so by Monday, the toxins that my kidneys hadn't cleared out of my system had gotten to me, and it made me sick. And the reason it made me sick is because my kidneys had stopped functioning. So I started throwing up, and the reason I was doing that was because all these toxins were running through my system. I tried to run, I normally ran 4 races, and I ran 3 of them, and I did very poorly because, well, my sympathetic nervous system had kicked in for 4 days. So they took me to the hospital afterwards. They thought I had the flu. When I got there, of course, they found out that my kidneys were not working. And so they kept me in the hospital. My kidneys, of course, started functioning again as soon as my sympathetic nervous system, as soon as the parasympathetic nervous system kicked in. Because I was, laying in a hospital bed, my wife was apologizing for being, the strange person that wasn't going to be running in any track meet anytime soon. They kept me in the hospital for a week to make sure that my kidneys, that it wasn't a permanent situation, that I didn't have a serious problem. So they diagnosed me with what they called athlete's kidney. In other words, the hyperadrenalinism, the fact that my sympathetic nervous system had been firing over a 4-day period had caused me to have kidney failure, as weird as that is. Now, one of the positive things, here I am 73 years old, if I really had bad kidneys, then something negative would have happened by this time. I haven't had any problems with my kidneys since, except I had kidney stones when I was 28. That was a long time ago, so I guess I'm okay. Anyway, there you go. So there's an example of the sympathetic nervous system overstimulating an individual, and their kidneys stop functioning. Now, normally when you have a situation like that, you would just sit down and you'd let yourself recover. You would try to force your parasympathetic nervous system to take over again, and that would have solved everything, except I needed to get rid of all the toxins in my body that the kidneys hadn't been able to get rid of because of the sympathetic nervous system firing over a 4-day period. The average human brain weighs about 1,400 grams. That's about 3 pounds. The major part of the brain is the cerebrum, which is divided into 2 hemispheres. Intellectual capacity seems to be contained in the surface area of the brain. The surface area of the brain is increased through ridges and folding that triples the surface area of the brain. The convolutions are called gyri and the furrows are called sulci. The brain contains 4 fairly distinct lobes that have 4 fairly distinct functions. The frontal lobe, this portion right here, spatially right there, prefrontal cortex, is in the front of the brain and is the seat of higher level thought. Now, if you think about it, if you play football, of course, you are constantly slamming your forehead into things, into the guy across from you, and that is not a good thing. If you are somebody that has been abused, a lot of times the forehead is the area that takes the pounding. And if that's the case, then this is the area of your brain that you don't want to hit. Well, you don't want any of your brain to be hit, but if someone is abused, then often the frontal lobe is the one that takes the brunt of the blows. But that's where your higher level thinking takes place. So if you've been hit in the head a lot, and especially in the front, it may cause an intellectual deficit to occur. The parietal lobe is on the top of the brain. This is the parietal lobe right here, the yellow portion, and it controls your fine body movements. The occipital lobe is in the back and controls sight, and this is the occipital lobe. And the temporal lobe is on the side and controls your hearing. If an individual lost half of their brain before they reach puberty, except for some fine motor movements, they would seem perfectly normal. And this does happen that people will lose half of their brain. The reason has to do with epilepsy. Sometimes people will be epileptic. Usually, epilepsy is only on one side of the brain. And in that case, if it was severe enough, they would do what they refer to as a hemispherectomy, and they would take out whatever half of the brain, whichever half was causing the problem. Now, what would happen is that the other half of the brain there's enough connections in the other half of the brain that it would make up for the half of the brain that was lost. And we've seen this over and over and over again. By the time they're 12 years old, of course, then by the time they reach puberty, the brain starts to be hardwired at that point. And you can't really do a hemispherectomy after puberty has started because the brain has started, and they will lose functioning if they do a hemispherectomy at that point. But if they can do it before puberty starts, then the individual will recover almost completely. The two hemispheres of the brain are connected by a mass of thick myelinated neurons called the corpus callosum. And the corpus callosum becomes very important. This is about 11% larger in females, so the connection between the two hemispheres of the brain is thicker. It's more complete as far as in a female's brain than it is in a male's brain. Now, the average male's brain tends to be a little bit larger, but the corpus callosum is smaller. If we cross-sectioned an individual's brain, we would see that some of the brain is made up of tightly packed, non-myelinated neurons that look gray, and that's on the surface. Other areas look white because of the myelinated neurons that make them up. Researchers have found that the larger the gray areas of the brain, the more intelligent the individual is. This is where your thinking takes place. Gray matter, non-myelinated neurons do things slower. And for that reason, intelligence has to do with not doing things fast. Otherwise, the intelligence would come out of the myelinated area. As the baby begins to develop from ever-expanding numbers of cells, the brain and the central nervous system begin as a structure known as the neural tube. In the beginning, the smaller, more primitive parts of the brain develop before the intellectual portion of the brain, the cerebral cortex. The spinal cord, the medulla, the pons, and the cerebellum all develop first. The cerebellum surrounds two areas that the scientists consider an evolutionarily old part of the brain that they sometimes refer to as the reptilian brain. And this is the reptilian portion that they're talking about. There are two portions of the reptilian brain, the basal ganglia. Basal ganglia means basic nerve sheath, and the basal ganglia is here. And the limbic system, the border system. Limbic means border. And it's in the middle. The basal ganglia includes the caudate nucleus, which means tail, the putamen, which means stone. Putamen looks like a stone. That's the purple thing. No, it's not. It's this part right here. Sorry. The globus pallidus, the pale sphere, and the substantia nigra, which means black substance. And there you go. All the structures of the basal ganglia. The limbic system is made up of the amygdala, the almond, which means almond. Where is the amygdala? There it is. That's it right there. The hippocampus, which means seahorse. And this is the hippocampus. This is the seahorse. The fornix, which means arch. And there is the fornix right here that's connected to the mammillary body and the hippocampus. The cingulate gyrus, which means round girdle. And this is the cingulate gyrus. the olfactory bulb, and it's not on here. This is the olfactory bulb. Wait a minute. It doesn't have it on here. The olfactory bulb in the mammillary body. This is a mammillary body right here. The largest organ in the center of the brain is the thalamus, the inner chamber, which means inner chamber, and it's right in the middle. The thalamus acts as a connector between the upper and lower parts of the brain. The real controlling gland in the brain is the hypothalamus, and that's below the thalamus. There it is. There's the thalamus right here. The hypothalamus is called the hypothalamus or lesser thalamus because it is below the thalamus. Thalamus is here. Hypothalamus is here. In reality, this tiny portion of the brain controls your hunger. It controls your thirst. It regulates your temperature. It controls reproductive behavior. It controls the pituitary gland, which controls almost all of the body's hormone secretion. And there's a pituitary gland right here. We used to think that the pituitary gland was a master gland, but the reality is that the hypothalamus controls the pituitary gland. Two of the most important areas of the midbrain are the tiny bumps known as superior and inferior colliculi. Colliculi means trough. The superior colliculi receives visual information. The inferior colliculi receives information about sound. The substantia nigra, the black substance, releases dopamine, and the substantia nigra is the area that is, if it's destroyed, if it has a problem, there it is right here. It causes Parkinson's disease. So a decrease in dopamine, a decrease of dopamine producing cells in the substantia nigra results in Parkinson's disease. Now, an increase of dopamine, if you have an increase of dopamine receptor cells, then that can cause a condition known as schizophrenia. So too little dopamine causes Parkinson's disease. Too much dopamine causes schizophrenia. The reticular means network, formation runs from the midbrain into the medulla. This portion of the brain controls sleep, arousal, temperature regulation, and motor control. It is the reticular formation that arouses a person when their baby cries. The cerebellum is composed of tightly packed and folded neurons. Now, one of the reasons I'm having problems with some of these figures, some of these figures, is because everything is right on top of each other. And what they've done is a cross section, and sometimes they show the organ that I'm looking for, and sometimes they're not. They don't show the section that I'm looking for. A good example is this. See, you can see there's two halves of the brain. We have two thalamuses, thalami. There's just about two of everything in the brain. So if you're looking at a cross-section of it, sometimes it's difficult to find them. Oh, I seem to be having trouble with it. So the cerebellum is composed of tightly packed and folded neurons. The neurons in the cerebellum forms an interesting fan-shaped structure that includes granule cells, Golgi cells, and Purkinje cells. Purkinje cells are contained in the middle layer of the neurons in the cerebellum. Purkinje cells are large, multipolar neurons shaped like fans with many dendritic spines. These cells run from the surface of the cerebellum to the brainstem. And this controls your movement. And this is one of the reasons why you can dance. And that's what a Purkinje cell looks like. The intricacy of the Purkinje cells allows the cerebellum to control both the fine and gross motor functions of the body. The position of the cerebellum between the spinal cord and the thalamic centers that communicate with the motor complex allows it to control motor function. And this is why you can drive a car. And those of you who can use a manual transmission, that's why you can push in on the clutch, change your gears, and release it as you're accelerating. So you have to do two things at the same time. You have to push in on the accelerator and back off of the clutch in order for the car to engage properly. And this is moving your feet in different directions. Not the easiest thing in the world. Unless you've done it a lot. And then it becomes natural. But it's your cerebellum that allows you to do that. The cerebellum leads into the pons, the first portion of the brainstem. The pons, which means bridge, is involved in motor control and sensory analysis. Information from the ear first enters the brain in the pons. The bottom of the brainstem is made up of the medulla, which means marrow or middle. The medulla controls both the neck and the tongue muscles. The medulla also contributes to the regulation of breathing and heart rate. Very important portion of the brain. Okay. Should I show you this? Well, we don't have it. Maybe. No, we don't. Okay, I'm sorry. It's going to talk about wrecking on your motor, falling off the back of your motorcycle. But let's go ahead and skip that. Maybe we'll talk about it later. The main intellectual functions occur in the cerebral cortex. That means external layer. The cerebral cortex is made up of gray matter. It has 6 layers. It's 6 layers thick and contains between 50 billion and 100 billion neurons. The functions of the various regions of the cerebral cortex have been mapped and divided into 46 distinctive areas known in toto as Brodmann's area. The most numerous neuron in the cerebral cortex is the pyramidal cell. Pyramidal cell dendrites reach to the surface of the cortex and also spread out horizontally. These neurons seem to be arranged in columns. Now this gets really interesting. Many brain regions have distinctive geometric columnar patterns that seem to function as information processing units. Some columns begin at the surface and extend all the way to the white matter. The human cerebral cortex contains about 1 million cortical columns. Most cerebral communication runs vertically, but there are some areas of horizontal communication. The cortical columns of the neocortex are arranged in 6 distinct layers. However, most communication is vertical, and select neurons will extend through several layers, some through all 6 layers. Now, this is how we remember things. Because of these columns, we need to get into this area to remember the information that we're looking for. Now, if you are one of the, am I going to talk about schizophrenia? No, I don't talk about schizophrenia. Let's talk about schizophrenia for a second. You can see how organized all of this is. This is very organized. People with schizophrenia, when they do autopsies and they look at these columns, they see them as all skewed around. A normal person will have columns that are vertical, and there is some degree of horizontal movement of information going from one place to another horizontally, but most of the information goes vertically. And if we looked at these columns for an individual suffering from schizophrenia, they would be all yin-yang. They would be all skewed around and not in a vertical pattern. And this is one of the reasons, if you've ever talked to somebody who had schizophrenia, they jump from subject to subject. They jump from, and sometimes they will say nonsense words. And this is one of the reasons why, because as I'm trying to speak, and I don't seem to be doing a very good job of it, but as I speak, I'm speaking in sentences. I'm trying to make sense of all of these aspects, and I'm using this columnar structure of my brain to get all this information out. But if I were schizophrenic, I wouldn't be able to do that, because as soon as I came up with the word aspect, then that I would start talking about insects. I could start talking about insects because the connection just wouldn't be there. And that's one of the reasons why people suffering from schizophrenia have such a difficult time dealing with reality, because their reality is all yin-yang. It's all skewed around. It just doesn't exist like it does for everybody else, because their columns are not vertical. As weird as that may seem. Besides the bony skull, the brain is protected by 3 protective sheets called meninges. Meninges is Latin for membrane. I think it's Latin. It could be Greek, I guess. The outer layer is a tough envelope of cells called the dura mater. Dura means mater, of course, means mother. Dura means hard, the hard mother. And as we know before, as we talked about it before, it's the astrocytes that make up this protective layer across the top of the brain. And if you've, well, of course you've never even seen any brain surgery, I don't know why not, but if you'd ever seen brain surgery, one of the things that happens is they have to remove a portion of the skull, and then they have to cut through the dura mater, and they have to use a scalpel to do that. And sometimes it's really tough, and they have a difficult time getting through it. Of course, the scalpel is very sharp, so they do get through it. I'm not trying to indicate that you know, they have to punch it up or stab it real hard. It's not that. It's just that it's very hard. The middle layer contains an open portion known as the arachnoid, through which flows the central, the CSF, the cerebrospinal fluid. CSF stands for cerebrospinal fluid. The inner layer is a delicate membrane known as the pia mater, or gentle mother. And of course, so there are 2 actual layers of astrocytes. One of them is the pia mater, and the other is the dura mater. Both of them are protective of the individual. Now, if the CSF gets infected, if there's an infection in the CSF, if a bacteria makes its way into the cerebrospinal fluid, the dura mater and the pia mater will start to swell. And the reason it is swelling is because it's trying to protect the brain. Now, if the swelling becomes too extensive, then it can do damage to the brain, and this is not good. And this is what meningitis is all about. A bacteria has gotten into the cerebrospinal fluid, and because of that, there's swelling in the brain area, and that is causing the problem. And it can cause the individual to die. And this is one of the things that we inoculate children against is meningitis. So one of the shots that your baby is getting is to combat meningitis. This has been a really serious problem for a very long time. They used to call it brain fever. And it was brain fever because you had all this swelling taking place. But before antibiotics, it was a death sentence. But now, of course, we can treat them, but we have to treat them with the right antibiotic. And I have a horrible story about that, but I'm not going to go into it. Sure I will. Okay, once upon a time when we were stationed in Germany, we had a baby that came in with meningitis. So you can treat them with antibiotics. The problem is you have to treat them with the right antibiotic, or it's not going to kill the bacteria. So what we needed to do was identify the bacteria that was in there. Well, the bacteria that was in there was a very delicate bacteria. So we had a sample of the cerebrospinal fluid so that we could identify the bacteria, and the Army had a sample of it. So we tried to identify it, and I was the one in charge of the microbiology at that point. So we put it on a plate of chocolate. I know that sounds silly. Well, we called it chocolate because it's brown. It was actually red blood cells that were hemolyzed, so it looked chocolate. It was broken red blood cells, and that fed the bacteria. That's the protein that fed the bacteria. So we grew it the first day, and we got some pretty good growth, and so we needed to identify the bacteria. It was a very small bacteria, one we hardly ever saw. And so the second day we were able to isolate it and identify it. So we identified it as Neisseria parainfluenzae, and which is rare, which is really, really rare. You see Neisseria influenzae far more frequently than you see the parainfluenzae. So we identified it as parainfluenzae, and the Army identified it. They had the same specimen that we did, but they identified it as Neisseria influenzae because it's more common, and theoretically they were growing that bacteria. So the argument they were life flighting these kids back to the States because there was no hospital in Germany that could help them. So they were life flighting them back to the States, and by this time 2 other children had come back down with meningitis, the same meningitis theoretically that the first child had. And so they started treating them for, that was the argument. Which one is it? Is it influenzae or parainfluenzae? The Air Force had identified it as parainfluenza. I had identified it as parainfluenzae. The lab officer at the Army hospital, which was a larger hospital, they had much more sophisticated equipment than we did. They identified it as influenzae. The doctors got in an argument as to which it was. The Army doctors won. And so they treated the child for influenza, for Neisseria influenzae. They treated the individual with a antibiotic that killed influenzae, but it didn't touch the parainfluenzae. And unfortunately, I was right, and the Army was wrong, and the baby died. The first baby died. When the first baby died and didn't respond to the antibiotic, then they started treating the children with an antibiotic that actually knocked out parainfluenzae, and the other children were saved, but the first baby did not make it. That is the sad thing. That's the sad story that I have about cerebrospinal fluid. They had to draw some spinal fluid out of the baby. It was not an easy task because there's not a whole lot of room to do that. We had it first. We were ahead by about 12 hours, and we identified it as one thing. They identified it as something else, and maybe they had contamination. I don't know, but we were right and they were wrong. So, and that's what happens in medicine. And you know, I was talking to somebody this weekend. You know, if I make a mistake when I'm working in the laboratory, it could mean life or death for somebody. And of course, you know, that type of responsibility really gets to you after a while. Anyway, and some people couldn't handle it. I seem to handle it okay, but other individuals had problems with it. That sobers you up really fast. The brain is not a solid mass. There are 4 open areas called ventricles, and this is where the ventricles are, right in the middle of the brain. As you can see, they're open areas. It certainly looks different from one angle than it does from another. The ventricles 1 and 2 are lateral ventricles in each hemisphere, and these are one. This is 1 and 2. The 3 ventricles, the 3 ventricle is between the hemispheres below 1 and 2, and this is it right here. That's the 3rd ventricle. The 4th ventricle is in front of the cerebellum, and this is the 4th ventricle, obviously, right here. Okay, and this is where the cerebrospinal fluid flows through these areas and around the brain. This is filled up with cerebrospinal fluid, the open areas of the brain. Now, the interesting thing is that people suffering from schizophrenia, they lose brain mass, and the ventricles keep expanding from the inside out. So these ventricles become very, very large. One of the things that we have discovered, since we are able to look at brains using MRIs and other PET scans and whatnot, because we're able to look at the brain, we can see now that people that box or people that have a lot of blows to the head, it causes a lot of damage. And this area of the brain, this open area of the brain, actually becomes larger. As the brain dies, it fills up with cerebrospinal fluid. And we know that now. CSF is produced in the choroid plexus, which means separate braid, a portion of the lining of the ventricles. Cerebrospinal fluid is basically plasma, or blood with the red and white blood cells removed. The function of the CSF, this cerebrospinal fluid, acts as a shock absorber for the brain. The brain floats in cerebrospinal fluid, so head movements do not disturb the brain. It collects nutrients from blood vessels and passes them on to the brain's surface. And that's the job of the CSF. Blood is supplied to the brain through two significant arteries that run on each side of the esophagus. Of the carotid, which means to plunge into sleep, the carotid arteries, and the vertebral arteries. The vertebral arteries bracket the spinal cord and enter at the base of the skull. And there you go. There they are. There they are, there they are, there they are. At the base of the brain, the carotid and basilar arteries join to form a structure called the circle of Willis. And this is the circle of Willis right here. The merging of these two cerebral arteries provides a backup in case blood flow is impinged in one of the two major arteries. So if you had a blockage in one of your arteries, your brain wouldn't die because this would kick in and you would get more blood flow from here. Despite the rich tissue of the brain and the myriad of viruses and bacteria seeking entry, infections in the organ rarely occur. This is because the capillaries in the brain are much smaller than the capillaries in other parts of the body. It's really kind of interesting because if you're talking about meningitis, you're talking about really tiny bacteria. And this is one of the reasons why it was so difficult to identify them. Bacteria are microscopic anyway, but the Neisseria bacteria is a really, really small bacteria. And so that's one of the reasons why it was able to get into the cerebrospinal fluid of the babies that I was talking about. This is, okay, substances in the blood will rarely pass into the brain. This is called the blood-brain barrier, and that's the fact with bacteria and viruses. They can't get into the brain normally unless, of course, you have an infection like meningitis. An angiogram, a vessel picture, is an X-ray of the blood vessels. And as you can see, this isn't what an angiogram looks like. If a stroke is suspected, an individual can be injected with a dye, and the skull can be X-rayed to show possible hemorrhages, aneurysms, or occlusions. An aneurysm is a widening of the blood vessel, and an occlusion is a blood clot, which causes blockage. And you don't want that to happen in the brain because then it kills that portion of the brain. This is usually the first test performed if a patient is suspected of having a stroke. Now we have a medication that we can give them if it is a blood clot that will dissolve the blood clot, with keeping them from damaging the brain as much as possible. A CAT scan is an X-ray of a thin sliver of tissue. If a patient is suspected of having a tumor or a stroke, a CAT scan would allow a physician to visualize the affected area. And this is the affected area right here. This is actually a stroke. This person has had a stroke. Areas, whereas the angiogram and the CAT scan are both X-rays, the MRI uses radio waves and other magnetic energy to visualize the structures in the brain. Small changes in structure, as small as the myelin on individual axons, can be visualized. Not in this picture, of course, but in other pictures. PET scans are machines that detect radioactive activity in the brain. The radioactivity comes from radioactive impregnated glucose injected into the subject. As the various areas of the brain metabolize the glucose for their activities, the researcher is able to see which areas are active with select tasks. And this is an area that is the most active because it's red. The redder it is, the yellower or redder it is, the more active it is. This is a person on cocaine. This is a person watching a nature video. This is a cocaine video of an individual who is a cocaine addict, whatever. A functional MRI differs from an MRI machine in the intensity of the magnetic signal used. Because of the functional MRI is so powerful that it can detect minute differences in metabolism. This gives researchers a similar picture as the PET scan, but without the radioactive injection. And as you can see, we have tumors. This is a tumor right here. This is the tumor post-surgery. They've taken it out. And this is the area where your speech comes from. Oh, okay, they took it out. Okay, this is an individual who has lost a limb, and they're still feeling their amputated portion, and of course, this is a pre-amputation. This has to do with pain, and this is afterwards. They still have to do with pain, but even they think that they feel pain, and that is the end of the chapter. So we will talk about chapter 3 next time, won't we? Yes, I think we will. Anyway, okay, I'll talk to you guys next week.